All right, so here's what we're gonna do is I'm kind of gonna take you through an example. I used to work on lung cancer, so obviously I'm gonna use that as an example. But this is a paper I did a long time ago. I did in collaboration with some people that actually work in this division on this floor. And what they wanted to do is, they had a target and they came to me and they said, hey, we want to find out what's going on. So what I'm going to do is I'm going to talk about that work, and what I'm going to do is frame it as a crime mystery, right? And trust me, there'll be lots more references you're not going to get through this. It's going to be awesome. So we're going to frame it as a crime mystery, and that way you can kind of see, I think it's a very easy to follow example of to kind of go through, that's how I feel, is like I'm an investigator. Somebody comes to me with a problem, I collect a bunch of data, I go to the crime scene, I look at all the different stuff, and then, I go, that's your subject, that's your, criminal right there. Okay, so to set this up, we're gonna set this up as in the middle there's a bank, right? There's a bunch of people walking around the bank, some people are in the bank, these are just, but here's what happens, the bank gets robbed, right? As soon as the bank gets robbed, in about 2 minutes, the police start rushing in, or maybe 5 minutes, the police rush in and basically lock everybody down so no one can move, okay? Where would you think most of the criminals would be that were involved in the bank robbery?
In the perimeter of...
Yeah, closer to the bank, right? Oh, by the way, you're an investigator. I used to use Columbo. No one knows who Columbo is. Do you? Sweet. Well, now you get SUV. Is she SUV or is that Law and Order? I can't remember which one she does. I don't know. Okay. All right. So here's our investigator. So yes. Okay, if the police run in, and lock everybody down, most of the criminals are probably going to be by the bank. But as you can see, and these are in red. You can see maybe there was a really fast criminal that ran, farther away. Maybe one had a scooter nearby and got a little farther. But for the vast majority, most of those criminals are going to be about around the bank. Okay, here's what we can do is we can draw perimeters around the bank. And this is kind of what we do with statistics. These are p-values. Everybody understands what a p-value is, right? I bet you don't really know what a p-value is, but anyway. But this is what we do with the p-values. It's like drawing a perimeter around the bank, right? I can draw one at, say 0.01, and basically most of the individuals that I have in there are probably going to be guilty, but we're absolutely going to have some false positives, right? We're going to have some innocent bystanders that were just walking around the bank that didn't have any, that weren't involved in anything, right? And we can make this perimeter as big as we want, right? We can bring it out and get more guilty individuals, but here, what is the trade-off there? As we expand the perimeter, what happens?
More false.
What? Oh, shoot. Yes, more false positives. As we expand that perimeter, the more false positives, which kind of muddies up your interpretation, right? You don't want to spend a lot of time on an individual, grilling him for 3 hours when he had nothing to do with the bank robbery, right? Here is what most people do in statistics. And instead of drawing it really wide, what they do is they use false discovery rates. Has anybody heard of a false discovery rate? You should. Because here's what happens is that I am studying, when I look at, a particular disease, humans got about 25,000 different genes in them, right? Every time I do a statistical test, there's a bunch of error. There's some error in that. As I do more and more calculations, I compound that error. So at the very end, say if I'm looking at 10,000 genes and I have a p-value of 0.05, how many genes would I expect to have just due to just random chance?
5%.
5%. 5% of 10,000 is what?
I'm not doing that.
I think it's 50, isn't it? 500? No, it's 500. 500, yes. Here is what happens sometimes, is a lot of people, and so p-values work for individual genes, right? Everyone in all of these different, individuals have its own p-value. What an FDR is, a false discovery rate, is basically what you're doing is statistics on an entire gene list. So I'll analyze somebody's experiment, and they'll say we're doing 10,000 genes, and they get like 425 that are significant. Does that bode well for that person's experiment? No, that's horrible, right? They're below the error value. So in a way, and I'll tell them, I'm like, listen, the amount of significant genes we generated from this is basically lower than what you would expect with error. So therefore, there's probably nothing in here. And to convince people of that is very, very hard because they spent so much time, energy, money. No one wants to admit that something doesn't work. But with statistics, you can absolutely kind of prove that. So again, that's what most people do is they'll draw, can you see oh, cool, all right. So they'll draw the perimeter very close to the bank so that what they'll say is, I want to draw this perimeter so close that I only expect 10% false discovery rate. Does everybody understand that? And you're almost assured that you're going to get guilty individuals, right? But you're also missing all these other genes or criminals that were probably involved in the bank robbery. Does everybody understand that? When would you think you'd want to, say, use like an FDR? When would you want to be absolutely certain that your criminal is who you got? Yeah.
If you want to pin down maybe a singular gene that affects a pathway.
Yeah, or what I would say is if you're looking for a biomarker.
Oh.
Right? Say I have somebody, I want to make sure that person that this gene, I want to make sure this gene absolutely changes in this disease because I'm going to use it as a marker of the disease, right? I would say I would use the p-value of 0.01. I would go out farther if I'm just trying to understand the system. There's lots of different ways, and the way you apply your statistics, here's what happens is these statisticians, they spend their whole careers developing a statistical tool, like a little hammer. And they spent so much time and energy on that hammer, they're going to use it to hit everything. Screws, nails, pieces of wood, they don't care, right? But what you really want to do is you want to have a toolkit of lots of different tools and apply the correct tool to what you're doing. And again, if you're looking for a biomarker, you definitely want to make sure that what you're looking at is true. So you would want to use the false discovery rates. Okay, so we can do that. So again, you are the investigator, and I forgot to tell you that you also have access to all the police databases. You can check Scotland Yard if you want to, anything you want. Okay. So what else would help you determine whether this individual is guilty or not, right? We already decided that those individuals closer to the bank have a greater chance of being involved in the crime. What else would help you? Yes. Yeah.
If they've done, if they have a history of big robberies in the past.
Absolutely, right? Criminal history, right? If this person, hey, of course, everybody's saying, I was just walking by the bank by myself, I'm not guilty. But if this person, you're interviewing this person, you look up and he's been involved in 3 other bank robberies, do you think it increases his chance he's involved in this crime? Totally. Absolutely. Right? And some of them, you might find that, that is the thing. If you've done criminal history in the past, you're more likely to do it now. And this is what happens a lot of times, the usual suspects. Does anybody know this movie at all? Okay, cool.
Is that Scar? No? Yeah, I don't think so.
Kaiser Sose. Yeah, exactly. It was a great movie. Too bad Kevin Spacey ended up being a weirdo, but still a great movie. I highly recommend it. So again, we're looking for the usual suspects, right? And here's what happens is, think of cancer, right? There's not just one kind of cancer, right? You have colon cancer, lung cancer, all kinds of different cancers, right? But say I study a lot of different cancers and I find out the same suspect just happened to be walking by the bank when it was robbed in all these different, say, cities. Does that make the odds that he is guilty go up? Yes. And we totally do this in bioinformatics. Remember, we know what these genes are. We can basically, if a gene pops up, I'm looking to see, all right, what has that gene been involved in before, right? You know, and here's the usual suspects we're usually looking for in cancer, right? It's the oncogenes for sure, right? These are the criminals. What are some examples of some oncogenes? Anybody know? Yes?
We have some KRAS.
Yep, absolutely. What else? Anybody know some other ones? Yep.
Is P53?
Is that an criminal?
No, that might be a is that a tumor suppressor?
Yes, totally. Anybody else? Any in your labs? What are you guys working on? Are you working on an oncogene? Do you know if you're working on a gene that is actually basically promoting the generation of the tumor or the cancer, or are you looking at something that's trying to prevent it? Do you guys know that? I mean,
Semaphore 7A promotes cancer.
what's that?
Semaphore. It's a protein? Oh. There's a gene that makes semaphore 7A.
Cool. All right, and then we're actually, keep in mind that gene. We're going to use that. So oncogenes, some typical oncogenes. You can say specific ones, but really a lot of them are growth factors, signaling molecules, right? Cell cycle genes, absolutely, especially cell cycle, inhibitors. Angiogenesis, if you're going to grow a tumor, it needs a blood supply. So angiogenesis is huge. You also have to protect the telomeres, right? If you're going to have uncontrolled growth, you got to make sure that you're adding things back onto the end of the chromosomes, right? And again, KRAS, EGFR is a good one, MYC, VEGF, right? So on the other hand, you have your oncogenes. That's just one part of the puzzle. Then you have your tumor suppressors, right? These are the cops trying to prevent the robbers from corrupting the system, right? So what are some tumor suppressors? And if you don't know like any specific ones, you can tell me classes. What do you think a tumor suppressor would look like? Yes?
Something that regulates like apoptosis.
Absolutely. Something that promotes apoptosis, which is absolutely P53. And in fact, I would say P53 is probably your biggest cop in your cell, for sure. Like he is the commissioner of all the other cops in the cell for the most part. But that's what you're looking at. Cell cycle inhibitors, DNA repair enzyme, BRCA1, BRCA2, telomere maintenance, programmed cell death, like we said, apoptosis. Some examples of that are P53, BRCA1, retinoblastoma 1, and P10. Okay? What I'm going to tell you, and here's what happens, is people study cancer and they only focus on one side or the other. And what you really need to do is you need to focus on the interaction between both of those, because it isn't just one or the other, right? It's the oncogenes trying to blind the tumor suppressor, and the tumor suppressors trying to find the oncogene, right? There's this tug of war here, and that's what you really need to understand is what does that battle between these things look like? And what happens is one gets more powerful than the other and suppresses the other, right? And then you either have cancer or you don't have cancer. I will tell you now that pretty much everybody in this room has had cancer. Why don't you think we're all dead? Yes?
DNA repair mechanism.
DNA repair, absolutely. And our immune system, for sure. Our immune system takes care of that before it becomes a problem. Again, another kind of suppression. So you've got the bad element. Here's another thing about the oncogenes. Are these genes inherently bad? No, right? They serve a purpose. They're not, when you say oncogenes, it's not like they were created to give us cancer. They perform a function, but just like in real life, right? Maybe they're in a bad neighborhood. They got really bad friends. If you're smoking, if you're doing things bad to your body, you're mutating lots of genes, right? You're causing the creation of these criminals. You're creating that bad environment for them to develop. Okay, so I'm going to talk about one criminal, that I worked on was Polo-like kinase 1. Anybody hear of that gene? Me neither. I didn't hear about it until my collaborator came to me and he said, hey, listen, we're studying lung cancer. We see these lesions in these, in the lungs. The ones that actually go on to get really, really bad have, really high levels of Polo-like kinase, but we have no idea what the hell it does. Can you tell us? Can you help us kind of figure this story out? And so I'm like, heck yeah, right? So first thing I do, look at the criminal history. What does this thing do, right? It's involved in cell cycle. It triggers the G2 to M transition, so it's stimulating the cell cycle. It's involved, it's localized to the centrosome, so it's probably involved in separating the chromosomes. And then the loss of this expression can lead to pro-apoptotic pathways. So you get rid of this, and then, basically the cell kills itself. So it's preventing apoptosis. And then, it can inhibit transactivation and pro-apoptotic functions of P53. So again, this criminal is fighting the biggest cop in your cell, right? It's the opposite of that. And again, the biggest thing is its expression is elevated in many different types of cancers. This gene has robbed lots of banks, lots and lots of banks. I have a feeling it's also robbing the lung cancer bank. Okay. And we can see that. So this is a website I use. The really good websites you kind of have to pay for. I'm sorry. But there are some good ones out there, especially the European ones. Ones from this country are horrible. But this is just showing you, if you go to the far right over here, this is 100% of the samples located in each one of these classes of cancer, right? So this would be 100% of samples. If it's red, it means that gene's overexpressed, which is Polo-like kinase 1. And if it's green, it means it was underexpressed in those samples compared to the controls. Do you think this gene's involved in some bank robberies? Heck yeah, holy crap. Especially bladder. It loves the bladder cancer bank. Breast cancer, kidney cancer. This is a huge criminal here. Okay? We can even do a close-up on lung cancer. Again, we're looking here, lung cancer. Almost like 98, 97% of all the samples in this database have some kind of alteration of Polo-like kinase 1. Based on this work, like, I'm onto the right target, right? I know this is a criminal, okay? And again, I know it's a criminal and not, I would say, a tumor suppressor. Why do you think I would know that? Yeah?
Wouldn't it be a lot more green?
Yes, absolutely, right? If it's a tumor suppressor, you're probably going to see the expression of this thing go down, right? You don't want the cop in the room. You want to take the cop and put him way down, right? You want to suppress the cop. So in this way, this is a criminal. You're basically promoting this criminal. Okay. So what crime did this criminal commit? The worst crime possible, murder, right? Has anybody heard of a Kaplan-Meier curve? Yes? Anybody not heard of a Kaplan-Meier curve? Survival? You haven't? These are used quite a bit in cancer. Depending on how they're used, they can be really informative or really, really BS. I'm just telling you right now. But in this case, I would say this is a pretty good Kaplan-Meier. We've got a lot of samples here. But you can see in the purple, if you have low expression of Polo-like kinase, you're more likely to survive than if you have high levels of this protein. And today, when we go into it, when we start doing our independent study, I'll go over the Kaplan-Meier and I'll go to places where you can actually see this on any gene you want. Not only that, but I went into the criminal history, and just like most criminals, they're not born bad, right? Like they're developed. Like, what you can do is you can go in the childhood of this particular gene, right? So what we can do is we can basically, with a what is the tube they use to go down in your lungs to look at different lesions? Does anybody know what that's called? It's a camera. They put a camera in your lungs. They can see lesions, and what they can do is basically know where those lesions are and then basically test whether those lesions are going to eventually become cancerous or not. Right? Normal lung kind of looks like this. When you start to get dysplasia, so this is kind of the pre-signs of cancer, you have different thicknesses start developing, you have, the membranes start going all weird, then eventually what happens is you end up with cancer. But up until, like stage 6 here, we don't treat it for cancer, right? We have to wait until it is. But if you look at Polo-like kinase, the ones in red eventually end up turning into cancer. The lesions in the blue end up actually reverting back to normal. And you can see that those lesions with high Polo-like kinase activity are more likely to turn into cancer. Again, maybe we can use this as a biomarker. That if I come in and I sample, I take a lesion, I find high levels of Polo-like kinase 1, and even if it isn't cancer, maybe I treat it like cancer, right? This thing is huge. Studying genes can let you know a lot about what's going on. All right, let's talk about pathways. So again, we talked about proximity to the bank, probably a good indication of guiltiness, also criminal history. What else would you might want to know? Again, you got databases that you can look at. Scotland Yard, FBI, whatever you want. What do you think else would help you determine who's guilty or not? Instead of criminal history, how about criminal associations, right? So let me give this as an example. Say I'm in the police force. I know there's a gang out there. There's about 7 individuals, and we stop everybody, and how about in 5 of those individuals just happen to be walking by the bank at the same time? Does that seem like, is that odd to you? Yeah, right? Again, everybody said, hey, I was just walking by myself. If I see 5 members of a gang out of 7 that were near the bank, I would say, yeah, that gang's probably involved in this crime. And that's what happens that's what we're talking about, is these pathways, right? And these pathways, these gangs can be huge, they can be really cool, or they can be doing some really terrible things in your body, right? And no one knows who this is? You may name that band? Cool in the Gang?
Oh,
Your parents would have laughed so hard at that joke. Okay. What about that one over there on the right?
Oh.
Jackass, yeah. I put that in there just so I can record myself saying jackass. So you have gangs, right? You have all these different gangs. They can be different sizes. They can be different, strength, But here's what happens is most of these gangs share gang members. And so you'll find one in gang. It's not just in one gang. A lot of these, like P53 is in about 1,000 different gangs, right? It does a lot of different things. But that's what we do, is what I try to do is I basically go in there and I say, okay, if I find a criminal, chances are he's associated with a criminal gang that's involved in it, right? Usually it's not just one person robbing the bank. I mean, if you really want to rob the bank proper, you need lots of people. And that's kind of what your cell does, right? For you to do anything, it requires more than one gene. So what I'm going to tell you now is basically the underpinnings of pretty much all bioinformatics. And after you understand this, you're going to understand more about bioinformatics than most people out there. And it's very simple. The statistics behind this kind of pathway analysis stuff, very, very easy. And I'm going to explain it in this way. So say I do a study. I look at, say, 2 individuals with a particular type of lung cancer versus the other. And I want to know if this pathway, which I think is involved in my pathway, I want to see if this pathway is involved in the condition, right? So what you can do is you can think of these 40 members of this pathway, right? Think of them as green balls. Oh, and say, I get 152 genes that I think are tied to this particular type of cancer, right? Right? So say these 40, the 40 genes in this pathway are green balls in a sea of 25,000 pink balls, right? And what I do is I get 152 draws from those 25,000 genes, right? There's 25,000 balls. What are the odds that I pull out one green ball? I think that's pretty rare that I would do that. Not really. I mean, you could expect that. Like, if I'm sticking my hand in 40 out of 20, I mean, it would be odd, but it wouldn't be unlikely. What happens now if I pull out 3 green balls? Right? Now that's starting to get very unlikely, right? The odds of me pulling, if I only have 40 green balls in a sea of 25,000 pink balls, me pulling out 3 is pretty rare. But this is usually what happens in bioinformatics, is say I pull out 7 balls, green balls, out of 152 draws, right? That doesn't happen by random chance. That is not an accident. Given that result, I would say that this pathway is probably involved in the condition that I'm looking at. And I don't have to get all the gang in there, or I don't have to get all the members of the pathway. I just need to get enough to show that that doesn't happen by random chance. Does everybody understand that? Okay, you know more about bioinformatics than a lot of people out there. And that's what I did. So I went, I looked at things that changed due to histology. So as the cancer got worse, these genes changed. And then I looked at, pathways, these gangs associated with, if that lesion eventually turned into cancer or actually regressed into normal. And what you find is when I start looking at the gangs here, the gang of, Polo-like kinase is definitely in there. Not only that, but it's really high, especially in the persistent group. Right? And this is a negative log p-value. So the odds of me getting this is, 10 to the 7.7, negative 7.7. So found my criminal. Did some criminal research on him. Definitely lots of criminal activity. Looked at other banks. He's been associated with those robberies. By the way, the criminal could be a she too. Okay, so that, and then now what I did was I go, okay, is his gang also involved? And I looked here and absolutely his gang is involved in this. And that's what you can do. I can pull that pathway out, and what I can do is I can basically color all the genes in this pathway with red that are also correlated to histology. And as you can see, lots of different members here. Here's the coolest thing is, once I know it's gang, maybe say Polo-like kinase is holed up in his hideaway, right? I can't get to it, right? So, but by knowing the gang, say I know that this SLK induces Polo-like kinase, maybe I rub that out, right? Take away its food supply, its support system, or over here, Pt2, maybe I get rid of this, and then, basically we can, stop the cell division. That is what's important. It's not just the gene. If you understand the gang and the environment it's in, you can figure out other ways to modulate that pathway. Does everybody understand that? All right, specialized networks. So these are things, so say you got all these individuals in the yellow that are by the bank. They're really close. They're probably involved, but say most of them don't have a criminal history. And you're like, what's going on here, right? But here, say this individual, these 3 work in the same auto body shop, and this is this person's sister, and this is this person's cousin, right? Not a criminal association. I didn't know that this exists, but say, now, we see relationships there, right? Like a party of individuals that get together to do, perform a particular function, right? Like a bridesmaids at a wedding, right? Anybody see that movie, by the way? That's a funny movie. I really like that. Okay. So yeah, specialized networks. And that's what we can do with string. We're going to work on this today as well. We're going to go through that. But what I can do is I can use this program to basically say, hey, in your database, I want you to pick out all of Polo-like kinase 1's best friends based on the data you have in there. And we can do that. And once I know its best friends, I can say, okay, what are their best friends doing? Well, it's all about cell division here, right? Which also allows me to understand the gene a little better. What we can also do with some of these programs out there is what I can say is I'm going to take all my genes that change And what I'm going to do is this program's going to insert other genes that may say aren't on my gene list, but say connect a bunch of things. So in this case, I used Ingenuity Pathway Analysis. Has anybody heard of that program? Yeah. It's very expensive. Some of you will probably never use it because it's so damn expensive. As you can imagine, some of this software is very, very expensive because there's a lot of data that it correlates to. So my advice to you is if you really want to do bioinformatics, get into a lab that's well funded, for sure. You can also learn programming, but that's not my gig. Okay, so we basically create this network. What happens is I find another individual here, VEGF. It did not show up on my gene list, but it's previously known to induce Polo-like kinase, and it also interacts with a lot of other things. So again, I found my criminal, I found its gene, I basically found its pathway that it's in, and now what I'm doing is I'm finding other collaborators. And now what I have is I think I found the boss of Polo-like kinase. Right? Master regulators. This brings us to master regulators. So I'm not going to ask you, here's the situation. Say I've got 5 individuals. They don't talk to each other. They don't know each other. But say they all take orders from one person or one gene. The Godfather. Do you think the Godfather was anywhere near the bank when it was robbed? Heck no, right? Yeah, I was in the bank. I wasn't by the bank. I was in a movie. He's got an alibi, right? He's the Godfather. And that absolutely happens in our body. Here's what happens when you have cancer. It's not just the genes that are near the cancer, but genes that are actually, say, producing growth factors at other parts of your tissue, right? In your bloodstream. You can have effects from other things that you can't measure its expression at. And that's what happens, P53 is it's a big time Godfather. If you actually look at measuring P53, you almost never see changes at the expression level. How does P53 do its activities? Does anybody know? No? Yeah.
It detects if when we're doing DNA replication, it is a fault.
Uh-huh.
It will detect that it's a fault and kind of send other genes maybe TP53 to go and maybe shut it down and
Yeah.
let it replicate, or maybe just.
Well, how do you think it does that? How does P53 get activated? It's all right. I appreciate you trying. So here's what happens is the really important stuff in your cell, right? Like P53. If I need to go somewhere quickly, am I going to call a cab or am I going to build a car? What do you think? Call a cab, of course, right? And your body feels the same way, is that if I have to basically make P53 from scratch, right, from the actual gene, it's going to be too late in order for it to do what it's needed to do. So what happens is your body, what it does is it makes it pre-made, and then it binds to say an inhibitor. Does anybody know what the inhibitor to P53 is? MDM2? Does anybody heard of that? Maybe a little bit. Okay. But what happens is you phosphorylate P53. What happens is, say there's DNA damage, there's a phosphorylation cascade. You phosphorylate P53, it disassociates from its inhibitor, which is in the cytosol, and then it gets translocated into the nucleus, right? You have activation, you have increased P53 activity without producing any more. Does everybody understand that? So the big things in your body, the really important pieces, you're probably never going to measure it changing at the transcript level. But with bioinformatics, we can get to these godfathers, right? It's like the RICO cases. I can get enough information on all its criminal associates to implement the godfather, right? So in this instance, I've got interleukin 1A in the middle. In this instance, it didn't change in my system. But what happens is everything that's connected to it, all these arrows, interleukin 1A is known to interact with, all of these genes. If there's an orange bar, it's known to induce expression. And if it's red, it means that gene went up in my condition, right? So you're seeing a lot of this like orange, interleukin 1A is known to induce this gene, and sure enough, it goes up, right? Also, interleukin 1A is known to inhibit certain genes, right? And that's indicated in the blue lines. And then if that gene went down in expression, you're showing the green color, right? The only ones not like showing a more active interleukin 1A are these 4 genes here, right? We got 43 targets of interleukin 1A, and basically, 39 of them are showing a more active interleukin 1A. Based on this information, I would say, even though I can't measure interleukin 1A, I know it's playing a role in my system because all of its criminal buddies are there. And not only that, but all their criminal buddies are going a certain direction, right? It's all positive. So it's a positive interaction. We do this stuff, I use this stuff all the time. And there's the statistics on it. It's actually not very difficult statistics. But here's what we can do it on. It's not just genes, right? I can use microRNAs, right? I can know if a microRNA is there. Say we know microRNAs target a specific genes, and if I see a bunch of genes going down, that it normally targets, and I don't measure the microRNA, I could probably say it's there because I can see what the actions it's doing. Think of it this way. If I'm in a pond, right, and I have a rock in my hand, and I throw it over my shoulder, right, and I wait 1 second, and I turn around, I could probably find where I threw my rock because it's going to leave ripples in the pool, right? And I just go into the center. Everything leaves ripples in the transcriptome. Everything changes gene expression, right? It's not just RNAs, it's chemicals. If you take drugs, I'll probably know if I get a blood sample. And it used to be, back in the day when I first started doing this, you'd see I studied aging, you'd see young people and old people. And in the young people, I'd always find, I'd find a lot of cannabis signatures in gene expression, right? We know all these chemicals, they induce certain genes. I can see that in the DNA, even though I'm not measuring the amount of joints this person is smoking, I can see its effects in their bloodstream, right? Or drugs. By the way, anymore, you don't see that so much. I think actually the older individuals tend to end up taking, I see more of a cannabis signature sometimes in the older individuals than the younger individuals. But back in the day, it used to be definitely the young people. There was a clinical trial I got involved in, and what they were doing is what was the drug they were testing? I think it was I can't remember. It was kind of a lung drug, and what they wanted to do is, MDs are great at saving people, but if you ask most of them to plan a experiment, they're horrible. Just absolutely and utterly horrible. So this was a poorly planned experiment. So what they did was they had a bunch of individuals that were on this drug, and they can only be on that drug. And then they figured, okay, now we need a control group. So what they did was they just got anybody they could in the hospital that was kind of the same age, as long as they weren't taking that drug. So you had a bunch of people that could only take one drug, older individuals, older males. It was a pulmonary hypertension drug, now I remember. And then you had a bunch of old males that could take anything they want except the drug that group was on. I found a difference, but it was actually in the control group. What do you think I found in the control group that wasn't in the experimental group? Older males. One group can only take one drug. This group can take everything they want except that particular drug. What do you think I found in this group?
A lot more variability.
A particular drug that maybe older males are taking. Viagra, yes, thank you. Yes. And it was the worst meeting ever. I had to go in, and they must have spent hundreds of thousands of dollars on this clinical test. And I had to tell them, hey, your drug got overshadowed by boner pills. So it didn't work. And they're like, are you sure? I'm like, yeah, I'm pretty sure. Yeah, that was a bad meeting. But I say these stories to just tell you, everything leaves ripples. Like I used to do experiments with mice, and I'd always start it in the morning and then get at the end. And I would find that you could basically predict the time of day based on the gene expression of certain genes in the mice, right? You have these period genes, circadian rhythm genes. You know, everything leaves a mark. And if you really understand it, if you dive in and try to understand that, you're going to know more about your system than anybody. Okay, so we're talking about VEGF, and I'll try to finish up here. What time is it? Ooh, okay. So again, VEGF angiogenesis. I found the godfather, associated with all kinds of bad criminal activity, associated with cancer. I can't measure the expression of VEGF by itself, but what I do is I can find all its criminal associates. So there was 51 targets of VEGF in my genes that were correlated to the cancer. I can graph all that out. It kind of looks like a mess over here. Kind of looks like a mess, but here's what I can normalize all that, subtract the mean from everything, put everything on the same scale. Bam! That, I bet, is the activity of VEGF in my system. As the cancer gets worse, VEGF activity increases. Yes?
I have a question. So, is in these tests, if you're testing the VEGF, I guess, if you have the tumors spread throughout your whole body, is it like accumulated all into one, or can it like localize where the different tumors are in your body? Like, do they express? Does that make sense?
Maybe. Yeah, we can. There's certain markers that you can use. Certain tissues, and I do this all the time, certain tissues express certain genes and others don't. So you can kind of look for that to determine. Or are you talking about to find the cancer other places?
Sort of like if you took a blood sample and you found out that you had, say, breast cancer, just based on the genes that were expressed, would you also be able to tell if it had spread based on like this software just from the blood sample? Or
you could.
Just like,
there's absolutely markers that will indicate metastasis. So I think what you're talking about is like you take a blood sample. What they do is cancer cells will leach off into your bloodstream, and you can kind of isolate those. So what they could do is probably isolate those cancer cells and say, hey, based on the genes that are changing in this cancer cell, we think this thing is definitely moving around. And if you do find cancer in your bloodstream, it's probably spread already. So,
I have a quick question.
Yeah.
About the y-axis on that bottom graph.
Yep.
Is the negative relative expression just like downregulated, or is that inhibited?
I would just say it's relative to the mean. So that doesn't mean it's going down. It just means it's at a low level compared to the average expression over all samples. Yeah. Yeah, and I would say by looking at the criminal organization that's associated with VEGF and basically normalizing that, this is probably the best indication of activity of VEGF you're ever going to get, right? You're looking at all of its organization. You're just basically taking its organization and finding how much it's making, it's basically taking all this thing. So it's this kind of technique is very powerful. And again, it helps you get to things that are kind of invisible. It's like me, you can't see it, and then you take flour and you throw it in the air, and then it kind of settles on your target. And you're like, there it is. That's kind of what you're doing with this gene expression stuff. And again, we can use this, I can use VEGF, or at least the targets of that VEGF, to say as a biomarker that, hey, I found a lesion. We got really high levels of criminal associates of VEGF. Therefore, this is probably going to be cancerous. Let's treat now rather than wait later. And again, I can find the gangs now of VEGF. I can go in, look at data. What is VEGF in, at very high levels, prognosis is not good for VEGF. But honestly, that's what I'm trying to do. If I'm studying cancer, I don't want to just look at, I don't want to look through a little peek hole, right? I want to see the whole thing, right? I want to go in there, look at the data, and basically maybe start with a target, but expand out so that I can figure out, say, the criminal organization. Everybody's seen the FBI movies where they have the bulletin board, and they have all the pictures, and they have the arrows and stuff. That's totally what I'm doing with genes, the same thing. right? That's what I'm trying to figure out. How do these genes interact? You know, what's going on? Who's controlling who? And once I understand it, then I can basically come in and like say, just basically break up the criminal organization, right? Like, why would I want to like mess with a wise guy when I got the Godfather right here, right? That's usually what happens in cancer is you spend so much time with the wise guy, you don't realize like the brains are way up here, and you got to like, you know, get to that. But that's really what bioinformatics is about. It's not just one level. It's almost like a tool. You can start really big, and then you look at the information, and then it gets you to a certain point, and then you get to that point, you realize that there's another, then you go big in a different way. Basically what you can do is you can figure out, you can go to big to small to big again. But the important thing is to realize, and this is what bioinformatics shows you, is like all this stuff is connected. And you can't look at everything all at once, but you can absolutely basically, you know, find an area where you can understand things at a level where you can actually tell somebody or design experiments to disturb that. Does that make sense? Any questions? Yeah.
So when you're calculating the statistics like Fisher's exact tests and score, what software do you use?
Usually the website I use it on gives it to me. If you're a programmer and you want to do statistics, I would use R. Have you used R?
Yeah, too many times.
I know. I hate R. I don't use R. What I do is I buy statistical packages that help me do that. Has everybody heard of JMP? JMP? SAS, Prism, things like that. I would say here at this college, and no one really utilizes it, but there's like a JMP program that you can get for free here. Talk to somebody. It's basically an analytical program that you can use to analyze data. It works really, really well. Usually what I do is I get a program that does that for me. And usually I have to pay for it because the stuff you pay for works way better. I wish free stuff worked good. I really do. I'm a hippie at heart, man. I wish we could, free love and free data and all that stuff, but the man tries to keep you down. So, all right. Well, let's take a little break here, and then when we come back, and let's take a maybe 10-minute break, and then we'll come back, and then we'll start actually messing around with this stuff. And you don't have to listen to me. You can actually do this stuff yourself. Okay.